She specialized in ophthalmology in Germany, and afterwards she did the PhD for clinical sciences in the Semmelweis University in Hungary. And in addition, she got the qualification in human genetic counseling and got her habilitation in 2017, where she started at the Eberhard Karls University in Tübingen, where she's also a leading physician and a specialist ophthalmologist for inherited retinal diseases. And since 2021, she has also a similar appointment at the Semmelweis University of Budapest. And today she will be talking about the clinical trials for CNGA and CNGB using gene therapy. So the screen is yours, Ditta.
Thank you. First of all, I would like to thank everyone who is joining this webinar, especially Mirella, who invited me to speak today. And today my topic is going to be achromatopsia, which I've been working on for over a decade now in Tübingen mainly. And as a clinician, I will try to keep my talk from this perspective, from the patient's perspective, and talk about disease itself and the therapeutic perspectives we have in this kind of disease. As you and everyone knows, this is a very specific retinal disorder, which patients usually describe as an out-of-focus, overexposed black and white film. So you can kind of imagine how a patient feels like who is suffering under this condition. It's an autosomal recessive disorder, a very rare disease with a prevalence of 1 to 30,000. We do have the advantage in this disease that we pretty much know that it's a monogenic disease, and 90% of all cases can be solved genetically, and we do know 6 genes now which can cause achromatopsia. I just listed here all the 6 genes and their relevance in Northern Europe. This will be very important because CNGB3 is leading with 60% almost in this European region, followed by CNGA3. So most of 32% of all cases are caused by CNGA3 mutations. And the other 4 genes are responsible for 1 to 3% of all cases. I mentioned this is for Northern Europe because, for example, in Asian populations, CNGA3 accounts for almost 80% of all cases, and even in Israeli patients, this is the most important gene. So these epidemiological parameters also have to be considered when you're running a trial in your special clinic. Most of these genes, the first 5 genes, are responsible for some changes which are happening in the visual visual cycle of the cone photoreceptors. We pretty much know what these gene defects cause in the cone photoreceptor function. The last and most recently discovered gene, the activating transcription factor ATF6, is a bit different from these genes. This is a key regulator of the unfolded protein response. And this was the first description that it can be causative in a human disease as well. And clinically, it also shows an achromatopsia-like disease, or achromatopsia as you wish. Because it's a cone dysfunction, most of the symptoms are, of course, regarding to this, complete or incomplete color blindness, photophobia, reduced central visual acuity. What we quite often see by patients is nystagmus, and these symptoms occur after birth. Of course, not everything is tested after birth immediately, but these are the leading symptoms of the disease. Patients do describe that dusk or darkness is very pleasant for them because the light is very blinding. As we know, functionally, this is a stationary disorder, but we will see that there are some changes over time, not in the function, but more in the morphology, only very decent, but still some changes. Previously, a couple of years back, we did say that the anterior and posterior segments of the eyes are usually without pathology. In the era of OCT and adaptive optics, we do know that there are some subtle changes in the retina. And according to the OCTs, which we can see in our patients, we can categorize them actually, beginning from the very mild category where you don't really see a change in the OCT results, and then also some changes in the photoreceptor outer segments. There is a hyperreflective zone, and even an intraretinal bleb can sometimes occur, or in very rare cases in some patients, retinal atrophy can also be seen. So we do know that these mild changes are there, and if we move on and do also adaptive optics imaging, we do see that the cones are there, but the cone mosaic is also altered in comparison to normal patients, so that there are photoreceptors, and there are these cone photoreceptors, which can be our target for any therapeutic intervention. However, they are altered a bit, so this is what also has to be considered. On the next slide, I'm just doing shortly the functional results. This is an ERG recording from an achromatopsia patient, where we do see that there are no recordable ERGs under photopic conditions. However, ROG responses look pretty nice, and they are quite normal as well. So there is a big question, which is everybody was talking about in the last recent years, whether achromatopsia is an ideal candidate for gene therapy. And we do have to consider that it has a lot of advantages. First, it is kind of a disorder, a stationary disease, so we do not have a progression, which is a great advantage. We do know the genetics quite good, so 90% of all cases can be genetically diagnosed, which is also a very ideal case. And we also know that there are some photoreceptors we want to treat which do not get lost over the progression, because there is only a very slow morphological change happening over time. So we would say that this is an ideal candidate for gene therapy. And actually, there was also a lot of work done in the last 20 years. In 1998, Susanne Kohl described the first achromatopsia gene, and ever since there have been a lot of preclinical studies also, and now we can say that we have a lot of work which was done preliminary. Small and large animal models for achromatopsia do exist. For example, CNG-A3 knockout mice, or even naturally occurring mouse, dog, and sheep models also exist, where we can just study the natural course of the disease, and also do even therapy trials as well. Also for CNG-B3, we have the deficient mouse and dog models, where we can get the informations we need if we are planning a human trial. And animal models also exist actually for the other less frequent genes as well. So gene supplementation therapy has also been done in preclinical studies, and the very first report was on a successful delivery of AAV vector-mediated GNAT2 therapy in mice, where they did see an increase in cone ERG responses and an improvement also in vision-guided behavior. Actually, because it's a very rare causative gene, more and more the focus was put on CNG-A3 and CNG-B3. And Stelios Michalakis already described it in 2010, that an AAV-mediated CNG3 supplementation shows cone rescue and in the ERG in a mouse model. And we do have here the ERGs for the rods, which stay stable over the treatment, and some enhanced responses here in the mixed responses, the standard scotopic flash, and of course the cone responses improved significantly in the ERGs in mice. They also tested how it has an effect on the behavior of the mouse models, and they did see that that there was some first evidence of sufficient plasticity of the visual system, that this newly one information for the brain could really be used for the mouse to improve their function as well. They also described that normalization of CNGP levels in cones could be observed and delayed cone cell death. So there was great hope, and there is great hope for further therapy trials that this could work out nicely. And even in bigger animal models, like in the sheep, we did see similar results that the ERGs show objectively that there is a cone response which can be recorded after treatment. They actually tested mouse CNG-A3 and human CNG-A3 delivery as well, where they both can show that there is an improvement of cone responses in the ERG, and also behavioral tests were very positive, and they could see and they could detect that the animals did this maze passage where they had to go over obstacles that with the treated eye, there was a significant improvement for them. This study was also very important for further planning, because it did show that there is a significant and long-term improvement of cone function. And there is a robust rescue effect, which could be seen up to 6 years now following only a single treatment, so that there was only one injection in the eye. And this is a very important data that after 6 years, it was still working. There were also AAV-based gene supplementation in CNGB, 3 mouse and dog models also. There was an observation that the success rate of cone therapy in dogs was promoter and age dependent. So they have tried to combine the gene delivery with the treatment of intravitreal CNTF, which allowed a successful recovery of cone function. And of course, there is sufficient data already in the literature on biodistribution, safety, and toxicology in mouse, dog, and monkeys also. So there is a lot of work done previously, so we could move to clinical studies regarding gene supplementation therapy in achromatopsia. Ongoing studies are listed here. Currently, there are 5 studies, and Tübingen was participating as the RDQ consortium in doing CNG-A3 achromatopsia gene delivery. So I'm just going to report on this specific study in the future slides. We have done recruitment for patients with CNG-A3 gene defects and achromatopsia. And from these patients, 9 have been chosen to participate in our trial. We have done an escalating doses of AAV8 CNG-A3 delivery. The first 3 patients have received the low doses, and after the first observations, there was a committee who was deciding whether we can move to the intermediate dose. And if we got a go, yes, then we move to the intermediate dose, and then in a similar fashion, the high dose group has also been treated. There's, of course, there was subretinal injection, so after vitrectomy, a classical way, so to say. You can see on these images how we planned the delivery. This small X on each slide shows actually the retinotomy site. The smaller circle, the smaller white circle is showing the initial bleb with only water solution, buffered solution, and then the bigger circle is just showing the treated area. We always elevated the fovea in these conditions, so cones in the foveal area have also been treated. The first observations were, because it was a phase 1 and phase 2 safety and efficacy trial, the very most important thing was safety for us in the first round, and we can report that there was an excellent safety and no real adverse events have occurred. What we have seen after the treatment, that there was a subtle hypopigmentation around the retinotomy site, which you can see here and on these images as well. And there was a hyperpigmentation where initially the bleb was, it is what we have observed, but there were no other functional problems or anatomical problems. On the OCT, because we elevated the fovea, it was important to observe how it reforms, and we can say that after 3 months, the foveal dip recovered completely. CRT, central retinal thickness, stayed very stable and over time, and we did not see any bigger atrophies regarding to the bleb itself, regarding to the treatment itself. In the functional results, I am showing you now results after 3 years, because we have already approached to this. And we can say that there was an improvement of the central visual acuity, 4.6 letters, and a contrast sensitivity improvement can also be observed, and a slight improvement in color contrast sensitivity, so that there is a slight improvement on function. However, you might ask, why not more? I think it's because of so many different factors which can influence the treatment efficiency, and most importantly, we do have to talk about amblyopia and how the brain plasticity changes with age. So we do say that you have to take and consider, even if the visual system, the photoreceptors are restored, whether the newly these new functions which we are winning through the therapy, how the brain can deal with that information. So there is a lot of work still have to be done. For example, functional MRI investigations are already discussing this topic, and we also have to consider how much we can win in adult patients who do have this deprivation in the cortex. We have also focused on quality of life, and actually developed a quality of life questionnaire only for this patient group. And they reported after treatment that there was an improvement in the ability to fixate, to recognize letters, as well as in the photophobia. So we do have hope that that could work. But in conclusion, we have to say that more patients are needed. Age might not be a good predictor of the disease state and visual function, because morphological results might also have a very important and a crucial part when choosing patients for this gene therapy trial. And we can also say that functional improvement, which we've just discussed now, is hindered by central deprivation. So if we try to move more to the children and young adolescent age, so the younger ages, we might see a more pronounced treatment effect in achromatopsia, and the treatment can probably work better if we are overcoming these obstacles done by amblyopia. So that there is a light at the end of the horizon, and I hope you have a picture now from what is happening in achromatopsia, and I'm really happy to welcome any questions. Thank you.
Thank you very much, Ditta. Very nice presentation. So I don't know if there's any question, but I can start because I have a couple of them. So you explained, of course, the trial where Tübingen was involved, and this was using a AAV8, but I was wondering if similar effects have been found for the AAV2 trial, or if they have been reported already, because of course we know that the AAV8 target really well the photoreceptors. Yeah. So I was wondering if it was comparable, or the virus could make a difference.
The very first results are available from our group, and other study groups still are publishing. So I don't really know whether there is a good comparison to that. But we see in the mouse model, which we have discussed in CNGB3, that there was an agent promoter dependency, and the combination with CNTF was very crucial for them, for their success. We are waiting for the long-term results also on other trials as well. Actually, these trials are still recruiting, and we have to just probably wait for the results and have more patients involved to be able to compare properly these different kind of ways of gene supplementation therapy in achromatopsia.
Yeah, thanks. And then the other question I had was a little bit about the construction of your clinical trial, how these vital doses were determined, and also the criteria to be these 3 patients for each each dose. So was random distribution, or there was any criteria taken into account?
Well, we have chosen the 9 patients. So we had a pre-treatment recruitment for all patients. We had around 40 patients, so we could who were interested and who did come, and we had all the results we needed to be able to decide on. And then we have chosen 9 of these patients. So the patients have been discussed in a very great circle with all the clinicians and all the research scientists, because it was the very first achromatopsia treatment trial done in human. And we didn't have any experience before that, and nobody. We were trying to choose patients from different kind of aspects, and because mainly it was a safety trial, but we also wanted to see some efficacy as well. It has been considered depending on age, how the patients are willing to participate, how much are we risking or not. So the very young patients, for example, were not in the first round really involved, because we are still learning together with all our patients. But from these 9 patients, what the doses is, that was non-randomized. So that we have also decided who has to come in these 9 patients. And regarding the doses, that was also a big consortium decision, which doses we should use. And 3 patients have been chosen because we did want to see a difference in these patients, but we didn't have the capacity to do 20 patients or so. So 9 or 10 was our limit, and we wanted to see some effect of the different doses as well.
Okay, thanks. So there's a question in the chat from Mirella, and she asks, would you expect L and M cones to be differentially affected, or there could be one of them primarily affected?
Good question. It's really, there was some consideration, and also in the mouse model, seeing that there could be some difference. Maybe there are some differences which we can see, but we still have to consider how we can, for example, in human, how we can see the treatment effect regarding objective measurements, for example, with ERG, because the treated area is so small. And if we are recording a Ganzfeld ERG, this will, I don't know if it shows or not. So we do have to consider which objective measurements can still show us whether there is a difference between the different kind of cones which are treated, whether we can see this effect.
Okay, thanks. So is there any last question for Ditta from anyone? No. Then I have a quick one on in the first slide you showed this mosaic of cones, and I was wondering if anyone has looked after treatment how it looks, if they eventually did redistribute or something. I don't know if
there are already some studies, but I also have to look it up if it's already published or not, because probably there are. It's an interesting question, and this would be nice to see. However, it is quite difficult, and we do have to accept that nystagmus is a great disadvantage if you do any recording in human, because of the multifocal ERG is not working, microperimetry is difficult to complete, and even of course for adaptive optic measurements, and sometimes even for OCT, you do have to take your time, and it is quite difficult to record it, but I hope there will be some experience on that as well.
Okay.
Thank you.
Thank you very much.